Career Academy. Today, students, yes, you can hear me? Hello?
Yes, sir.
Okay, then we will start. Uh, Today we will learn something about the plant growth regulator. We will solve some question and side by side we will learn something. Okay, this is our first question is that you are given a tissue with its potential for differentiation. Okay, potential for differentiation in artificial culture. That is in the culture medium. The tissue is there and it has the capacity to differentiate. Differentiate means that differentiate means it start forming new organ. Here it is differentiation is organ differentiation. Okay. This is one way. Maturation. This differentiation process is there in different levels: cell differentiation, tissue differentiation, and here it is organ differentiation. Means formation of new organs: a root, a stem, branches, leaf, flower, fruit. This is so. For this also, this differentiation, growth hormones has important role. So which of the following pair of hormones would you add to the medium to source secure shoot as well as roots? Okay, so this is for the shoot and root differentiation. We have to add the auxin and cytokinin in a combination in proper proportion, in a particular ratio that is required. Different plants tissues require different amount. Actually, this auxin, no, this initiate root. Auxin is for root initiation. So if you need that the root to form, then auxin is to be given, and cytokinin is for shoot system, shoot differentiation. Okay, so a balanced amount of auxin and cytokinin, if added to the culture medium, then the tissue will start differentiating shoot and root both. Okay, our next question is that phototropic curvature. Phototropic curvature, you understand that when in a plant, okay, in a plant there is light is coming from a source, okay, then what we find that the plant bend towards the source of the light, plant or the plant organ. No, this is what we call the phototropism, is it? Phototropism, and this result, this is phototropism is due to auxin again. All right, this is for this one is for auxin again. Auxin cause, it was You know that Darwin, is it? Darwin, Charles Darwin. Okay, Charles Darwin, whom we know more popularly, we know for the evolution, is it? But Charles Darwin and his son, Francis Darwin, they were carrying out one experiment on canary grass. Canary grass coleoptile. This coleoptile is the membranous cover over plumule. Plumule that differentiate into shoot system, and coleoptile is the cover of the plumule, but not in all, it is in the grass embryo. Okay, it is in the grass embryo. So this coleoptile, when the seed start germination, the embryo start growing. So naturally this cover also will grow. So this cover will grow, and it will, they were they were observing that what is the effect of light if the light is coming from one side. If the light is coming from one side, that is unilateral light, then what happened? They found that the this coleoptile was bending towards the source of light. Then they concluded that there is something, certain chemical, which can transmit and cause the bending of the coleoptile. So, and from there, of course, this auxin was discovered, and also discovered as that a certain chemical which has the capacity for bending of the plant organ. Okay, so induction of phototropic movement. This was later on, of course, another physiologist who is known as F.W. Went. He also carried out similar experiment, but that was carried out in Avena. Avena, that is oat with, there is a very good breakfast food, I think many of you may take. Okay, Avena sativa. In Avena coleoptile, he carried out the experiment. And that experiment is known as uh, Avena coleoptile, Avena coleoptile curvature test, or Avena coleoptile test. This was a very famous experiment because all these things lead to the discovery of the plant growth regulator, growth hormones. Okay, so here is the next question related to that. The auxin can be bioacid by this Avena coleoptile curvature test. Okay, Avena coleoptile curvature test was carried out. This was by, what is bioassay? Bioassay is the testing the potency or potentiality, potency of a chemical substance or chemical compound in living tissue or in the living system. This is not only testing their presence also. The presence of this bioassay test was done. So it is also, we call the bioassay of Auxin is bioassay of Auxin, AU. Auxin is by Avena curvature or Avena coleoptile test. Okay, carried out by this FW Went. FW Went also credited for the isolation or extraction of Auxin from this Avena coleoptile. Okay, so he isolated, identified that chemical and identified as a Auxin. Okay, Auxin also, of course, isolated from human urine. Auxin also isolated from human urine. Okay, this was first. There is a little doubt, of course, but it is in our NCERT book. It is given that the first it was isolated from human urine. Okay, we will go to the next question. This fruit and leaf drop at early stages can be prevented by the application of, again, this is Auxin. Okay, the star mark is given here. So this is that this in case of plant growth regulator and the animal hormone, there is some major difference is that animal hormones are very specific. Okay, animal hormones are very specific. Their effect is unique. Effect is unique. It is same. One effect it will show. But in case of plant, this is not the specific effect. One plant growth hormone, they can show several physiological effect. And again, in different plant, in different species, the physiological effect of the same growth hormone is maybe different. But in general, we have to see what is the general effect of that. So one thing is that it is Auxin, it prevent fruit and leaf drop. This is abscission. Okay, the formation or the dropping of the leaf, flower, fruits, the birds, etc. That is known as abscission. Actually, there is a process of detachment of the organ from the plant. So there is a leaf or leaf drop or fruit drop. This is so it can prevent, but it prevent, the auxin is peculiar, it prevent in the earliest stage. Say when the leaves are young, when the fruit is young, very young, that is in the earliest stage, flower in the bud stage, or they are all earliest stage only. But in case of mature those organ at the later stage, that is when a leaf is old, in that case auxin will promote abscission. That is during the young stage it protect, but at the mature stage it will promote that the leaf, the fruit, they should fall down. So this is at a different stage, same hormone is working again in two different way. Next is another question, that is the apical dominance is due to. First, we have to understand what is apical dominance. This is dominance, and dominance is apical. That is the tip portion, top part of the dominance of the top part. So this is actually apical bud dominance. Okay, this one in the plant growth is due to the apical bud. Say here there is a plant, bud is there, in the bud these are the leaves. So this bud keep on growing, and at the axil of the leaf there is a lateral buds, okay, or we call the axillary bud. And here is apical bud. This growth takes place. This apical dominance is that this apical bud will keep on growing and the lateral one will not grow. So this is suppression of axillary bud or lateral branches of or we can say axillary buds by with suppressing by the fast growing apical bud. So long the apical bud is there, from the lateral side the plant will not produce branches. So this is also one of the physiological effect of auxin. If you want that the lateral buds or the branches should form, then what you have to do? You have to remove the removal of apical buds, allow lateral branches to grow. So if you want in your plant, in your garden plant or any other plant, that many branches should be there so that the many flowers or many fruits will be there. So you can, what you can do, you can just pluck the tip of the plant, then you will find the lateral buds are growing. So by this way, you can make a plant bushy. So when you want a bush or you want that a haze, etc., you want to maintain, then this apical bud removal is there. That is the decapitation or pruning. Is that we do pruning? No? This is very much practiced in tea cultivation. By pruning in tea plant, repeated pruning in tea plant, they make the plant very much bushy and don't allow them to grow very tall. So in many cases, this is very much useful. Of course, in some of the trees, say like timber trees, we don't need the lateral branches. And that's why what happened whenever the lateral branches coming out, then usually the lateral branches are removed. Okay, so that only the plant tree will grow straight. Next question is that which of the following is not a physiological effect of auxin? So the physiological effect of auxin, which one is not? Several effect is there. We have seen this. What is we have seen that apical dominance. We have seen that prevention of the abscission. Okay, so here this is initiate rooting of stem cutting. We have initially, I have told that auxin initiate root formation, is it? So this is the physiological effect of auxin in those plants which are planted by their cutting, stem cutting, okay? In those plants, if auxin is applied, then the root formation process is faster. In most of the fruit plant, these fruit plants are multiplied by their cutting, so that they will maintain the genetic characteristic, okay, the quality of fruit, etc., from generation after generation. So this is also done in case of tea, different variety of tea. There is also by cutting it is maintained. So in the cutting, for faster root formation, this auxin is applied. Then next one is promote bolting. This one is not the physiological effect of auxin. This is the physiological effect of another hormone, that is gibberellin. Okay, this one is not. So our answer is which one is not the physiological effect of auxin. So this is our answer. Next one is prevent fruit and leaf drop at earlier stage, that we have learned. This is the effect of the auxin. Inhibit the growth of lateral buds. This is also auxin effects are there. Of course, physiological effect, there are several other, like it promote, other physiological effect are promote flowering. Okay, so if auxin is spread in plants, the plant will start flowering. Not in all. This thing is practiced a lot in case of pineapple. It promotes flowering in pineapple. This one then promote parthenocarpy. Parthenocarpy is the formation of seedless fruit. Okay, so this, of course, here also, as I told, the effect, the physiological effect of a particular hormone may not be same in the different group of plant. So it shows parthenocarpy in tomato and some other plant, but not in all. This help in the xylem differentiation. That is xylem tissue formation. Then one is peculiar thing is this it is weedicide. This is used as weedicide. One hormone 2,4-D. This is a weedicide. This one that it kills weeds. Okay, so a spraying of this hormone kills weeds, but it is selectively kills broad-leaved plants. Broad-leaf plants means dicot plant. Monocot does not harm monocot plant. So when we monocot are narrow-leaved, okay, this so in the say like we are cultivating paddy, all right, paddy, wheat, maize, and some other crops. No, these are monocots. So in the monocot, in this field, if the dicot weeds are there, then we can use this 2,4-D. There is another one, 2,4,5-T. Okay, their combination or mixture also sometime is this also can be used for the defoliation of tree. That is, in the tree, if you want that the leaf should fall, then hold a leaf, then this auxin can be, this hormone can be applied, sprayed. We have to know little more about this hormone. Okay, this type of auxin. Auxin may be natural and artificial or synthetic. Or synthetic. Okay, synthetic, that artificially, chemically it is synthesized, and natural one is synthesized within the plant. This is one is IAA. IAA is indole 3 acetic acid, and another one is IBA, is indole butyric acid. These are natural auxin, actually indole, no? So auxin is indole compound, which is synthesized from a precursor chemical which is known as tryptophan. From tryptophan it is synthesized. Tryptophan is an amino acid. Okay, among the artificial there is NAA. NAA is a naphthalene acetic acid, and others are that 2,4-D, 2,4,5-T. 2,4-D is 2,4-dichlorophenolic acid. 2,4-dichloro dichlorophenoxyacetic acid, and this one is actually 2,4,5 trichloro. Okay, trichlorophenoxyacetic acid. These are the artificial or synthetic auxin. Now let us come to the next one. Okay, now we have some question related to the hormone cytokinin. Okay. Cytokinin. The question is, here we can see two questions, that the hormone present in liquid endosperm of coconut. Coconut is, this is cytokinin. And also similar question, of course, coconut water is rich in auxin. As coconut water or the coconut milk, they contain the cytokinin in large quantity. So this sometime in the tissue culture also, this coconut milk or the coconut water is added in the nutrient medium for the differentiation of the tissue. Cytokinin is a cytokinin, as the name you know, that it is cytokinesis promoting hormone. This one is cytokinesis promoting hormone. Okay. So cytokinesis, and what is cytokinesis? During cell division, you know, During cell division, what happened? Cell divide. First nucleus divide, that is the karyokinesis. And after that, there is cytokinesis. Division of the cell cytoplasm. And by this cytokinesis, one cell divide into two. So this hormone initiate or promote the cell division, and that's why by this way it induce the growth of the plant. This was found very much rich in coconut water, and also one this cytokinin was identified, discovered as kinetin. Kinetin, and it is found in the places, in the region it is produced, or it is rich in tissues where a rapid cell division is there. Okay, so usually in the shoot apices, root apices, the young leaves and fruit and flower, where the division is very fast, in those places this hormone is found. This hormone, and one kinetin, cytokinin, which is known as G18. G18, this is found in the maize grain. That is in the corn. It is found. Maize is Gia maize, so it is called the G18. This is also a natural cytokinin. Of course, cytokinin, it was first discovered by Skoog and Miller from autoclaved herring sperm DNA. From the sperm DNA, it was extracted. Okay, this herring, amphibian sea animals, from that sperm DNA, it was discovered. It is autoclaved, means it was sterilized, and in that high pressure and temperature, of course, DNA break. Actually, this cytokinin is a derivative of is a derivative of purine, particularly adenine. Adenine, you know that adenine is a purine base of DNA, is it? So this is this chemical form is actually 6-furfuryl aminopurine. The cytokinesis, they promote here also promote, so already told that promote formation. This is enhance the promote the transportation of nutrient, and due to this that it can the organs of the plant where the cytokinin is more, the organ of the plant they remain fresh for longer period. So this hormone cytokinin they can delay the process of senescence. Senescence is that is the aging and death. Aging and death. Okay. So this aging process or the death process is a little delayed. This is a hormone for the plant. It is very, very important. So by spraying cytokinin, we can delay senescence, death of the plant or plant organ, even in the vegetable, etc., in the shelf or in the shop or in a store, if is to be kept fresher for few more days, then this cytokinin helps a lot. Now come to this is now come to another hormone, that is the gibberellin. This gibberellin causes what? Gibberellin causes apical dominance, flowering, internodal growth, and wilting. So here it is gibberellin cause internodal growth. See, all this auxin, cytokinin, and gibberellin all are growth promoters. They promote growth and development of plant in various ways. And for the growth promotion, cell division, cell enlargement, all these things are there. So gibberellin, what it does, that gibberellin promote the elongation of the cell. Elongation of cell. This is cell enlargement. You can say cell enlargement. So if the cell, if say in a particular tissue, there are 100 cells, is it? And they enlarge very much, then naturally the volume and size of the tissue will be more, bigger. So this gibberellin promote growth by enlarging the cell number. And that's why what happened with the application of the gibberellin, this internodes, plant's internode nodes increase in length. Plant become very large. Okay, these things, what happen if those plant in dwarf plants, which are genetically dwarf also. If there the gibberellin is used, they can grow tall. Grow tall by the application of gibberellin, even the dwarf plant, and particularly say like plant like cabbage, no? Cabbage, they are very short stem, crowded leaves, etc. are there. So their internode is very short. But in cabbage also, if this gibberellin is applied, cabbage plant also can grow tall like 6, 7, 8 feet tall also, or even more. So this can increase the growth, and that is what we call sometime when this type of plants having the rosette habit. Rosette habit. Many plant you have seen that they don't have almost a stem. Their leaves are very near to the ground. Visible stem branches are not there. Only the root and the leaves are there. They are the rosette plants. All right, like beets, etc. Also, they have the rosette habit. Carrot, see, they have root and then leaves. No, very short stem. So in those plant, there is the process that is known as bolting. Bolting is a prominent physiological phenomenon in which suddenly if plant branches, a stem, or the buds, they start growing very fast. Fast growth takes place. Usually we can see that after the winter also sometimes some bird start or the plant start growing very fast, and then they bear flowers. So this is called bolting, and bolting is by the growth hormone gibberellin. Gibberellin is gibberellin. There are many. This is actually gibberellic acid. Gibberellic acid, Ga. Okay, and in different plant, variety of gibberellin has been found. But there are more than 60, 70 gibberellins are there, and they are named as Ga1, Ga2, Ga3, Ga4. Out of this Ga3 is very, very common one. This was discovered in Japan by Kurosawa. Kurosawa, he observed one seedling, paddy seedling, okay, a rice seedlings growing very tall, but not bearing flower fruits. Okay, and so the no flower, no seed, fruit. So they were called as foolish seedling. Foolish seedling, or this is also known as Bacon disease. All right. Bacon, Bacon disease. So, and this was due to the causal organism. One fungi is there, which name was given, that is Gibberella. Gibberella fusicoroi. This is a fungi belong to Ascomycetes, right? And this was the causal organism for the Bacon disease. And so this was later on the chemical was identified as GA, gibberellic acid. All right. So Gibberella fusicoroi was actually the source of gibberellic acid. So internodal elongation is due to gibberellin. Next is hormone that promote rapid elongation of internode or petiole in deep water rice plant. Okay, this is okay. Let us know little more about the gibberellin. Gibberellin also can cause. It can promote seed germination. It can cause parthenocarpy. And it is stronger than Auxin. That is, Auxin also cause parthenocarpy in some plant, but gibberellin is a stronger parthenocarpic habit or effect. This can cause, this is useful in malting. It speed up malting in barley. It help in germination, actually break the seed dormancy. Malting, so it is used in brewing industry. It is used, also can cause early seed production and maturation. This also can delay senescence process. This one of the very important, so I told that is important that different hormone has a different effect on different plants, different group of plant. It delay senescence, that is aging and death. Okay, so what happen if in the fruit, apple like apples and other, if it is a spread, the fruit will large and it will elongate, And as it can delay senescence, so the farmer can keep the fruit longer on the tree. So this is this has a commercial value. No, we may not pluck all the fruit at a time. If it is longer, we can keep the fruit on in tree. This its a market value will be more. Okay, otherwise all the fruit will come at a time and we have to sell it at cheaper rate. So this thing help that by delaying senescence. This one, this can increase the increase sugar production in sugarcane. Okay, because by spraying gibberellin there in the sugarcane, the internode will be longer, means the sugarcane plant will grow very tall. They will have large cell. They will have a thickness of the plant will increase. Though so the naturally the sugar content also will be more. So they can increase like this way. Okay, next one is one hormone, another hormone that shows the internodal elongation. The hormone that promote rapid elongation of internode or petiole in deep water rice plant. Actually, the plant that is growing in water, sometimes they produce a gaseous thing, that is the ethylene. Okay, ethylene is a gaseous hormone. In this way, this one is very unique. This is the only one which is different. Others are solid and liquid, but this one is gas. All right. So this is this one. In the deep water rice variety that when it grow, actually the plant growing in the in water, their flower leaf should be above the water level. Okay, and there this ethylene can promote the faster growth of the internode and petiole. So it is ethylene. Actually, ethylene is one of the growth hormone that has different types of uh, physiological effect. It function as a promoter, it function as a inhibitor also. This one is promoter as well as inhibitor. This hormone ethylene has one. Here is a similar question. That one I have already told, that the which of the following is a gaseous hormone. Say this one is West Bengal JE 2007. This ethylene is a gaseous hormone. This ethylene is very important for fruit ripening. This was of course discovered also as in the store, fruit stores, fruit go down. that where one physiologist, Auxin, he observed that some volatile substances says from ripening orange induce ripening of banana. Okay, so it was, and later on it was found that it is a gas and it is ethylene. So ethylene cause, so this is also known as fruit ripening hormone. So it is popularly known as fruit ripening hormone. It also the promote flowering and synchronization of flowering. Synchronization of flowering. Synchronization of the flowering, that all the flower will come out, start developing almost at the same time. At same time, sometime in some of the crop, what is there, that if the flowering is together, then harvesting of the fruit or the crop is very easy. Otherwise, what happen if but in the same plant different times the flowers are coming out and fruit formation, ripening, it is in different time, then what happen, harvesting is a problem. You cannot harvest it on the same day, same time. So this one is very useful in the pineapple cultivation. Pineapple cultivation. Okay. Next one, ripening of the fruit is controlled by ethylene. So this, now which of the following generally act as antagonist to gibberellin? This is another hormone is there which is known as abscisic acid. Abscisic acid, in short we call them ABA. This is actually, this one is retardant. That is, it is mainly inhibitor. It inhibits the different physiological processes. Particularly, we have seen that auxin, cytokinin, gibberellin, they mainly their function is promotion, but abscisic acid, it inhibit all the process. That is, growth will be inhibited, flower formation, shoot formation, root formation, this seed germination, all these things are inhibited mostly. When it was discovered, this was discovered also as a like a, inhibitor. Okay. It discovered as a inhibitor. One was discovered as a dormine, that is which promote or suppress the seed germination, that is it promote dormancy. But this hormone is very useful in the sense that it can it increase resistance to plant for various stresses, say dry condition, drought, okay, no water, very high intensity of light and sun temperature, high temperature, etc. is there. So the plant will develop the resistance, and by developing resistance, they will survive, overcome the odd situation. And that is why this hormone ABA is known as a stress hormone. And in general, this one oppose antagonist to gibberellin. Whatever the physiological effect is shown by gibberellin, usually it opposes that. One of the important thing in ABA, we find that stromatal closure is due to this ABA activity. Okay. Which of the following acid is the derivative of carotenoid? This is also ABA. Abscisic acid is a derivative of carotenoid. ABA is the derivative of carotenoid. This gibberellin, GA, it is a terpene group of chemical. Cytokinin is adenine derivative. Now, one question here, you have to think a little bit. One hormone is used to speed up the malting process in barley. Another is used to promote flowering in pineapple. And the third one help to delay of leaf senescence, delaying leaf senescence. What is the combination? Say first, second, third, we have to see here. Malting process of barley, that we have told, that is the gibberellin, is it? Gibberellin is the hormone which increase or promote malting. So our answer may be either this or this. Then it promote flowering in pineapple. Promote flowering in pineapple. Out of this, in pineapple, promoting flowering is either ethylene or auxin. So ethylene is for synchronization. Here it is auxin. Second one is auxin. And third, delay senescence. This cytokinin delay the leaf senescence. So this one is the answer. Next question is that some statement given, which of the following statement is correct? The causal organism for fully seedling, I think, remember the fully seedling, these are for the that Bacon disease, GA or gibberellic acid, gibberellin disease is the source of gibberellin. Yes, this is true. Fullis seedling disease, causal organism, the organism which caused the Fullis disease. No, that Gibberella physicori, that one is the causal organism. So this statement is true. Abscisic acid is a growth promoter. Here it is say, this one, abscisic acid is growth inhibitor. ABI is inhibitor, not promoter. So this statement is not correct. Ratio of auxin to cytokinin control cell differentiation. The very first question that we have solved, that is, this is true. So this statement also is true. Bolting of cabbage can be induced by treatment with IAA. IAA is indole acetic acid. This is auxin, is it? But auxin does not cause bolting. It is the effect of gibberellin. Bolting is by GA, gibberellic acid or gibberellin. So which of the following statement is correct? Is R. So which one is correct? 1, 2, 3 are correct? No. Correct statement is first one and the third one. So our answer will be this, that first and third statement is correct. Okay, so these are few questions that some expected and important question from the growth regulator. So if you have any question, any doubt, please you can ask. Do you have any? Okay, then we will stop. We'll stop here.
Thank you, sir.
Thank you, sir.
Thank you.